Prior to beginning the activity, please be sure to review the faculty information and disclosure statements, as well as the learning objectives. After listening to the activity, complete the post-test by clicking the Earn Credit link in the episode description. Downloadable slides and resources are also available. The following presentation is copyrighted by Medscape. No use, broadcast, or recording of this presentation or any part thereof is permitted without the written authorization of Medscape.
The following presentation is part of a certified educational activity provided by Medscape Education and supported by an independent educational grant from UniCure.
Welcome. Thank you so much for joining us to hear more about the current clinical research landscape in Huntington's disease, or HD. My name is Erin Furst-Dimming, and I'm a movement disorder neurologist at UT Health in Houston McGovern Medical School. I'm honored to serve as the HDSA Center of Excellence at UT. I am thrilled to be here today with my dear friends and colleagues, Dr. Claudia Testa and Victor Sung. Dr. Testa is a professor of neurology and the founding director of the Huntington's disease program at the University of North Carolina in Chapel Hill. Dr. Sung is also a professor of neurology and is the HDSA Center of Excellence Director at the University of Alabama in Birmingham. Of course, both wear multiple amazing hats, but in the interest of time, we'll move on. So I'm going to give you a brief overview of Huntington's disease. I suspect this will be a review for most of you, but just to ensure that we're all on the same page. So let's get started. What is Huntington's disease? What is HD? HD is a relentlessly progressive, inherited neurodegenerative disease that's really defined by its symptom triad, which I'll discuss in a moment. HD is thought to be relatively rare, but we know that the worldwide prevalence varies. We currently say that there are roughly 41,000 individuals in the US living with HD, with over 200,000 at risk. However, we suspect that this is a gross underestimate for multiple reasons. We know that men and women are affected equally. Unfortunately, most individuals with adult onset HD become symptomatic during the prime of their lives, during their 30s or 40s. However, there's a wide range of age of onset, so it's important that we as clinicians keep HD on our differential when we see individuals with extreme ages with symptoms consistent potentially with HD. Juvenile onset HD is rare and a far more severe phenotype. So HD is inherited in an autosomal dominant fashion. It's caused by a pathogenic CAG repeat expansion in the HTT gene, which is located on the short arm of chromosome 4. 26 or less CAG repeats is considered to be normal. 40 or more is consistent with complete penetrance. We know that the mutant Huntington gene produces the Huntington protein, the elongated Huntington protein that we believe leads to neurodegeneration in HD. So here's the symptom triad that I briefly mentioned. We know that patients with HD will have motor, cognitive, and psychiatric symptoms. The motor and the cognitive symptoms will progressively worsen throughout the disease course. The psychiatric symptoms, however, are far more variable, with the exception of apathy, which will predictably worsen over time and can be quite problematic not only for our patients, but their care partners as well. HD, as we know, is progressive. Individuals are born with a mutation. There's a period where they are asymptomatic or pre-symptomatic. And then there's a period where subtle neurodegeneration begins, and individuals have mild cognitive and motor symptoms. And this period has historically been called the prodromal period, and is in theory a window of opportunity, an opportunity for us to intervene, not if, but when we have an approved disease-modifying therapy or multiple approved disease-modifying therapies. Hence the HDISS. So the Huntington's disease integrated staging system is a biologic, evidence-based research staging system that facilitates earlier enrollment into disease-modifying clinical trials. This is, of course, a slide we could spend the entire time talking about symptomatic therapy in HD. I want to highlight the fact that we have effective symptomatic therapies in HD. We have effective pharmacologic and non-pharmacologic to care for our patients with Huntington's disease. I can't underestimate the importance of our allied health professionals, speech therapies, physical therapy, and occupational therapy, in addition to our multidisciplinary team members that help us care for our patients and their families with HD. To date, we do not yet have an approved disease-modifying therapy in HD. However, we are cautiously optimistic about the research underway. So speaking of the research underway, let's talk about the approaches for the current disease-modifying trials. Most of the trials that are underway or soon to be underway are focused on reducing the level of the mutant Huntington protein and/or limiting CAG repeat expansion, somatic expansion, that we think drives onset and progression of disease. There are several therapeutic modalities listed here. You'll see ASOs, RNA interference molecules, small molecule splicing modulators, and DNA level editing and transcriptional repression. There are over 50 companies that are currently invested in disease-modifying therapy and research. It's an incredibly exciting time in the land of HD-related research. Today, we're going to focus on the top 4 on this slide, so I'm going to now pass it on to Dr. Testa to talk about the ASOs.
Thanks. So we're going to be talking about 2 antisense oligonucleotides, or ASOs. They're both working through lowering Huntington, RNA and protein. So antisense oligonucleotides are targeted at pre-spliced RNA, so coming off the gene into RNA, and then before that final messenger RNA is put together that then makes the protein. That's where ASOs are targeting. Tominersen, our first one, Huntington lowering, it's going to be what's called allele non-selective, so lowering both the mutant and the wild type, all of the Huntington products. The WVE-003 compound is allele selective. We'll get into that in a minute. So tominersen, this is one of their proof of concept slides. This is just showing mutant Huntington % change, but it does lower all forms of Huntington in a dose-dependent manner. So thanks to this and a lot of other research, the tominersen team ran a pivotal phase 3 study that was actually closed at the last pre-specified data safety analysis time point. And that's because of the separation between treatment and placebo. So here in this graph, what's being shown is change from baseline in the composite unified Huntington's disease rating scale. This encompasses motor, cognitive, and functional measures. So negative change is worsening. The black is the placebo arm. You can see that there's hundreds of participants in each group. The orange is the lower exposure, so a loading dose, and then 120 milligrams per dose every 16 weeks. And that trends down along with the placebo group results. The blue is the higher overall dose, a loading dose, 120 milligrams every 8 weeks. And there you see that group at the 69 weeks separating from placebo, but not in the direction we wanted to see, instead getting worse than placebo over time by the 69 weeks. So why did this happen? We fundamentally don't know. There's been a ton of really helpful detailed post-hoc analyses, a lot of them made rapidly public by the research team that has really informed both the tominersen current trial design and all the other trial designs that we're going to be talking about today. Again, you see this is a large number of people. This is an exposure analysis. So exposures, the milligram per dose, the frequency of dosing, and then a little bit about how individuals metabolize that dose to come up with a measure of exposure. In the highest exposure cohort, well, you can see in all of them, compared to placebo, the pink line that lowers is the CSF mutant Huntington. Again, it's going to lower all Huntington, but this is a direct measure of mutant Huntington, and that goes down in that dose-dependent manner. But in the highest quartile exposures, you see that red line, that CSF neurofilament light chain. You see an increase in the third highest quartile, and in that very highest quartile, you see that big spike We have come to understand that this is a safety signal to pay attention to in these disease-modifying trials. We don't have a direct through line of why this would happen, therefore worse outcome, but we've now come to understand that this is something to really watch out for. In contrast, if you look at the two lowest exposure quartiles, that red line compared to placebo, CSF neurofilament light chain, is about the same, or if anything, over time, even better, implying that in lower exposure quartiles, you're going to get potentially a good risk-benefit therapeutic window. The other critical post-hoc analysis to understand from this trial is separating the participants into groups by age and what's called CAP score, which is a measure of CAG repeat length, age, and predicted trajectory towards disease progression. I don't have a pointer, so if you're facing the slides, the upper left thing is the low age, low CAP. The gray placebo goes down. The blue, the every 8-week higher, still going down, but the orange, the every 16-week lower, is actually stabilized and potentially as good as or better than placebo. So putting together lower exposure quartiles and this focused trial population, the Tom Anderson team is running a new trial that should have data out in 2026. What's really exciting is that at this meeting, they came out publicly with some information from a very recent independent data monitoring assessment. So the clinical trial team is blind to the actual details of that, but they get recommendations from that data analysis. So there were some major recommendations. They have 3 arms: placebo, 60 milligram every 3 times a year, so every 16 weeks, and 100 milligrams every 16 weeks, no loading dose. Recommendations: placebo continue, 100 milligram group continue, 60 milligram group continue but convert over to 100 milligram doses going forward. No concerns on safety. So this is really exciting. To me, what this means is that this agent is probably going to have a fairly narrow therapeutic window in terms of both dosing, but the population of people living with Huntington's disease or carrying a repeat expanded gene. To make a clinical analogy, I think if this proves efficacy and goes forward to clinical availability, going to see similar opportunities and challenges as with the new disease-modifying agents we're all learning in Alzheimer's disease, where they're really restricted to early Alzheimer's disease. You're thinking about how to get people that diagnosis early, how to get the right population, that right treatment as quickly as possible while they're still in that therapeutic window. So in contrast with WVE-003, we just have some phase 1b/2a data to review, but it's also driving their new clinical trial design. So this is an allele-specific. Again, we're targeting pre-spliced RNA. And the way that they're targeting mutant only instead of wild type is leveraging the normal variability that we all have across our DNA. DNA has a lot of variability in it person to person, but a lot of it seems to be kind of like your fingerprints, where there's a lot of variability. We can certainly measure it, but it doesn't seem to have a clear or even any functional impact. The expanded CAG long repeat length mutant alleles tend to be on a background of this DNA variability that certain sets of what's called single nucleotide polymorphisms, or SNPs, SNPs. This is where one of the pieces of DNA is a little different person to person. You can leverage the SNPs that are in or around nearby a gene to target that background. If the person has that SNP pattern and they're mutant Huntington allele and does not have that pattern on the wild type, then you get allele selective targeting, allele specific targeting. This is just their proof of concept of lowering mutant Huntington as opposed to wild type. But I think the bigger news here, now that we have all this other data from the Thompson-Nursing trials and others, is the neurofilament light chain levels. This is a fold increase, so a non-linear fold change on the y-axis. The gray bar is where mostly the placebo arm of their study is with NFL, and the dark lines are individuals with active compound. They're mostly staying within the zone of the placebo, but there are some individuals with higher spikes. Why are those individuals having higher spikes? Does this have significance for their outcomes in the trial? The WAVE team is publicly talking about continuing to refine their SNP targeting to get even better, stronger mutant Huntington selective lowering, and continuing to refine their understanding of where do these NFL spikes come from, why do they only occur in some people, and how can we avoid them in these interventional trials going forward.
All right.
Thank you. On to you, Dr. Sung.
Thank you, Erin, and thank you, Claudia, for running us through those ASOs. The next section, we're going to talk about two different drugs. That's going to be AMT-130, which is a gene therapy, and Votoplam, which is an oral splice modifier. So let's talk about AMT-130 first. This is the compound that has really dominated the news feed and social media over the last few weeks. That is the big outcome that has generated a lot of the buzz. So just to break this down, remember AMT-130 is a gene therapy that requires brain surgery, 6 trajectories directly injected into the brain, one-time surgery. And there were 12 patients that made it to the 36-month follow-up for the press release, and those 12 patients are analyzed here. And in the 12 patients who received the high dose, they actually tested a lower dose and a higher dose. And the high dose seems to be really positive and showing some benefit here. And on the same composite UHDRS measure that Claudia introduced earlier, the 12 patients who received the high dose over a 3-year time period only progressed by 0.38 points. Again, this is still 0.38 points of worsening, but a very small amount of worsening, and they compared to an external control group that from the Enroll-HD observational data set that was kind of matched as well as they could to the patients who received the treatment at the beginning of 36 months ago. And those patients in the control group progressed by 1.52 points over 3 years. It's important to note that this is an external control group. The caveats here being there's not an internal control for a brain surgery delivered therapy. It's very difficult to have a sham surgery internal control. They did have 10 patients who had sham surgery as part of this trial, but they were unblinded at a year. So at the 3-year follow-up time period that this data was released, they don't have 3-year follow-up data on those internal controls, and so they felt that this was the best that could be done, and they did pre-specify this with the FDA. That's an important designation. They asked the FDA, hey, since we don't have internal controls, since all the sham surgery patients were unblinded, what's the best thing to do statistically? And this was pre-specified, and their pre-specified primary endpoint was high dose on this composite scale measure. And this was met because it is statistically significant. The secondary endpoint that was, again, pre-specified was the total functional capacity. This is one of the parts of the composite UHDRS measure, and in particular, this is a functional scale that's measured 0 to 13. And at the 36-month mark, the separation between the patients who received this high dose of AMT-130 was statistically significantly better than the progression of the external control group. And if you look at this over time, there does appear to be more separation over time. There is still, in the high dose group, some slight worsening in the total functional capacity score, but it's less than the external control group. So it was not statistically, this separation was not statistically significant at 24 months, but was at 36 months. And I think you can see that further separation over time. And then just the comment that generally regulatory bodies like the FDA care greatly about not just biomarkers, not just any clinical measures, but can drugs that we're trying to bring to market have a functional impact on patients, particularly in difficult diseases like Huntington's disease that have significant functional impact over time. So I think this is an important measure here. And then what about safety? So Claudia talked about NFL and NFL as an important safety biomarker. We would estimate that NFL in Huntington's disease throughout the natural course of the disease does increase over time in the 10-ish% per year range. And this is interesting data here. At least it's not going up. I think that's really important. Unlike some of the ASOs where we had some spikes in patients. So in this gene therapy where they had drug directly injected into the brain, these patients all had immediate, early, significant NFL spikes, but at the 36-month mark, those big spikes have now gone away, and all the patients, both in low dose and high dose, are showing NFL levels that are actually lower than baseline. I think some people are trying to claim that maybe there's disease modification here, but I think that's hard to say with such small numbers. But I think what we can say definitively is that NFL not going up is a good sign for something as invasive as a brain surgery. So now moving on to the oral splice modifier Votoplam. So these are oral agents. This was their phase 2 data that was released a few months ago over the summer. And at the 12-month mark, both HD-ISS stage 2 and HD-ISS stage 3 patients had a dose-dependent lowering of mutant Huntington in both the blood and CSF. So this is the Votoplam 5 milligram versus 10 milligram. And there is more lowering of mutant Huntington protein with the higher doses. There is a minimal difference in the lowering between the ISS stage 2 and stage 3 groups. And this is to be noted that this is a pretty broad range of patients if we include all of HD-ISS stage 2 and stage 3. So this is interesting. And an oral molecule splicing modifier is going to be easier to take than many of the other therapies that we've discussed thus far. But this is phase 2 data, not powered to show efficacy, but these are some of the efficacy numbers that they had in this small data. So this is important to note that there was an open label extension, so all of the placebo patients converted to active drugs, so they didn't have placebo patients at the end. So again, this is comparing to a natural history control, and there are some early trends that the same clinical measures that we've discussed previously are trending in the right direction for 5 and 10 milligram Votoplam. So I think this is good, not enough to reach strong conclusions about efficacy, but enough to move towards a phase 3 trial. And this is just to show that the NFL data also trending in the right direction, so going down instead of going up at the 1-year mark. So this is, again, strengthens the evidence at least to move forward to a phase 3 trial, which is being planned.
Great. Well, thank you so much, Drs. Testa and Sung, for those insights. I know we all feel like there's so much more to say, but trying to at least get some highlights in. So let's just talk a little bit about, we've talked about four compounds that are under study. We've just attended the HD Research Congress, the HSG meeting. I think we're all feeling inspired and invigorated. Do you mind telling me just a couple of the highlights, a couple of the take-home points that we can share with our friends today?
I think in terms of what we've just talked about now, immediately applicable in Huntington lowering, is that we're starting to really understand from the clinical trial data we're just talking about and the new pivotal trials where we're going to be getting data out in the next year or two, Targeting matters if we're targeting this area called exon 1, where the CAG repeat expansion is. If we're targeting somewhere else, exposure level matters. Although we're all coming from the US, sometimes more power is not better. And focusing on which populations within Huntington's disease have the best benefit to risk. You could really see the different clinical trial teams leveraging all the stuff we just talked about to try to accelerate all this. And then we didn't even talk about somatic expansion and all these completely different targets. Just wasn't enough time. And somewhat coincidental, the stuff that's kind of on the front line of the closest to hopefully getting into clinic is Huntington lowering. But all these other mechanisms, I think, are going to be really exciting.
Yeah, I think it's also important to touch on, I think a lot of our viewers, we spend a lot of time talking about the data, but I think a lot of our viewers are going to want to know timelines. Yeah. And I think just to touch on that for each of these 4 compounds, just to summarize those, like going top left. So tominersen, ongoing phase 2 trial that is in that low dose, low age, low cap group. We should have data readout in 2026. If that's positive, then they'll spin up for a phase 3 trial. Wave, they're preparing for their big phase 3 trial. They're going to launch that in early 2026. Votoplam, they're launching their big phase 3 trial also in early 2026. AMT 130, even though this is a 1B, 2B, was phase 2 trial, 1-2 trial. They are packaging this data up and are preparing a submission to FDA sometime in early 2026 also. So,
yeah, and we don't know what will happen. So those are the timelines we know.
That's right.
Right? But just like with the AMT 130, the expectation was this is a phase 1, 2A, very small numbers.
That's right.
And so we don't know what will happen with the FDA submission. The FDA might say, great, go for it. They might say, yeah, you got to do more confirmatory study work first.
Right.
So it'll be very interesting to see what happens. And similarly, if any of the other combos we've been talking about, if the tominersen current phase 2 gets extremely strong data, maybe they'll try for an accelerated approval instead of spitting up the phase 3.
That's right.
So I think it's a really dynamic timeline, but we've got a lot that we're expecting to hit and understand more in 2026 alone.
Yeah, the next year is going to be a really exciting time for us all.
Yes, absolutely. So I think we have a little bit of time, a couple of minutes for questions from our audience, which I believe may be popping up here. So let me just take a quick look here. Oh gosh, lots of wonderful questions. We just must have another session, but let's start with one of the first questions. How is wild type Huntington measured in these clinical trials? I know we talk a lot about the mutant Huntington protein levels, and are they looking at wild type Huntington protein levels in the CSF?
Yeah, so currently there isn't a direct measure of just wild type Huntington, so it's a subtraction. That's oversimplifying it, but you can measure total Huntington, and then there's ways to measure mutant Huntington or different Huntington fragments, and then you figure out wild type from the totality of the data.
Yes, thank you for that. And then some questions, not surprisingly, we all had these questions when the data was presented, going back to Dr. Sung to the AMT 130 data, and some questions about the 5 patients. There were 17 in total, of course, but there were 12 that were presented, and so asking about the differences in
what happened to the 5. So 2 dropped out of the study, and 3 have not reached the 36-month time point in analysis and would not reach that time point until middle of next year. And in discussions with FDA said, go ahead and analyze what you have instead of waiting 8-9 more months to package that together. So that was the decision that they made. But we should have data on 3 more of those high-dose patients reaching the 36-month time point next year.
Wonderful. And then questions just about how do I enroll a patient into one of these studies? Are there ongoing studies in Europe? And yeah, so maybe speak a little bit about just how do we get individuals that are interested in research plugged in. And I know you mentioned Enroll HD, but maybe that's kind of a nice place for individuals to start. And so just talking a little bit about access to clinical trial opportunities.
Yeah, Enroll HD is an observational study, so you don't have to be in that in order to be considered for interventional clinical trials, but it does plug you into information. The tominersen phase 2 is fully enrolled, and as is the Unicure AMT 130. So as more things come up and running, I track Huntington Study Group website, I track clinicaltrials.gov, or at any of our centers, the trial coordinator or the case manager is always happy to hook people up, whether they're going to be sending them to our center or just saying, actually, this other group is much closer to you. Here's their direct contact.
Yeah, I think that's one more important thing to emphasize with the things. We gave some of the timeline things, but we didn't say it explicitly. None of the 4 things that we discussed are actively enrolling or accepting new patients
correct.
now.
Yeah.
So, even though that we're getting 50 plus calls a day about these after this big news, we don't have any place to put these patients right now. None of these trials are actively enrolling, but stay tuned.
But
future
stay
is going to be
tuned,
very
yes,
exciting.
because you mentioned trials that are up and coming that
That's
will
right.
hopefully early next year. Well, thank you both so much for your insights. I think, as I said, we're all feeling inspired, invigorated, and most importantly, I think we can tell our patients and their families that there's reason for hope. And so we're so grateful that all of you listened to us today, and thank you again for listening to us today.